I was just starting out by saying that today we're going to be talking about a number of things, but one of the main topics is going to be analyzing a little bit more detail that fundamental gene regulatory network toggle circuit that we had I'd assigned as one of the homework problems. And so if you were able to get through that on your own, that's wonderful. If there was an issue with it, we'll have plenty of time here to explore it and work through it. As always, I do record these, and I don't know if anybody's actually looked at them, but they are, the edited versions are up on YouTube. So if you want to refer to the lectures after the fact, they're there. I'm going through this material a little bit faster than I have in previous years. I'm not doing that not to try to rush you or to leave out things that are of interest to you, but people seem to be more interested in the computational side of things than the mathematical side. And so I've tried to reduce the amount of pure mathematics that I'm doing in class and focus on the kind of computation that we can do. The mathematics is available to you in the textbook and elsewhere. And if you feel that it's useful to me to come back and cover some of that in more detail, I'm happy to. I have many, many lectures of worth of slides available on these topics. And so if there are particular topics that are of interest to you that we're skipping, that you feel would be useful, then by all means say so, and I can put that material back into the sequence of the course. But at the moment, the plan is that we're going to not go into the mathematical detail. There's a lot of material related to bifurcations and to dynamical systems theory that we're not going to go into in a lot of detail. We're going to use a little bit of it today. One of the reasons that I'm not going to focus on it is that the dynamical systems theory techniques, they really apply to all of these systems, but classically they're most useful when you are doing low-dimensional systems, 1D, 2D systems with one variable or two variables. When you start dealing with complex networks with many variables in them, then the dynamical systems methods are not as helpful. And so you get thrown back more and more than on numerical techniques. And so the other thing that I realized, and if you notice it, please call me on it, is that the printed copy of Herbert's textbook that I have and the electronic copy of the more recent edition are slightly different, even though they show the title page and the copyright information are identical. And so it's possible that some of the page numbers and chapter numbers I've had in the previous slide decks have not been right, because Herbert actually added material and reorganized the contents. So the topics are all there, but they're not in quite the same order in the latest version online as the printed version. And so the chapter numbers here at the bottom are correct with respect to the latest PDF version. And as you'll see when I say that we're jumping ahead a little bit, we've got chapter numbers like 12, 13, and 15. There's additional material in those chapters that we're not going to dig into in a lot of detail. And we're going to come back in the next week or two and sweep some of the earlier chapters. There are a set of chapters 9, 10, and 11 that are about parameter identification and fitting, and we'll come back and do those. One problem is that things like lectures are intrinsically linear. I have to give things in some order. But in fact, to do these problems, you need to come from many perspectives. And so there isn't some perfect way of introducing the material. You always have to do something first and then come back and sweep the ideas. So again, if you find these confusing or something is left out, then by all means ask me to fill in the details. Also, I've mentioned it before, there's that Mathematics of Life textbook that I think is a really great way of learning the basic mathematics if you want to do. So for today, I'd like people to give me a quick update on their project ideas. There was a homework problem that asked you to start clarifying your ideas. I don't know if you have had a chance to talk to each other about things. I really would ask you today that by the end of by tomorrow, I would like everyone to make a appointment for just, if it's just a half an hour Zoom session with me to discuss one-on-one projects. So that I'd like to do that this week. I understand we're still early in the semester, but I want to have a chance to talk to you, brainstorm with you a little bit, and help you refine ideas. And then we're going to do two things that are a little bit separate from the rest of the class, but are quite useful. One of which is how to download pre-existing models and run them. And then we'll talk a little bit about functional declarations in Antimony, which we've actually, I think, already used. And then we're going to jump into the toggle, which was one of the homework problems, and do some analysis. And I really would like, if possible, to get to stochastic modeling today. If we don't get to stochastic modeling today, then we'll do that next week. I wrote up a little homework problem set, which I've got printed out here, but it focuses on the stochastic models. So if we don't get to that today, I'll try to come up with the problem sets are always going to be essentially one or two simple problems and then working on your project. And so if we don't get to the stochastic modeling today, then I'll come up with a different problems for you. So maybe to start with, I'd like to hear from people about what maybe your homework 2 problem, so to speak, is. Who would like to tell us, maybe do a quick screen share and say where you are with your projects ideas.
I saw your email about the Luca and trying to model that, and I tried to like find any like information on that, but I think they just don't know. Like, I guess I saw they had a couple, like they kind of had stuff mapped out or whatever, but I couldn't find like data sets or something like that, so I didn't know what that ended up being.
That's okay. I realized I was way behind on epigenetics and a few other things. So I'm always telling people read Wikipedia. So I was reading Wikipedia articles, and the Luca article had a little bit of a metabolic cycle for sort of hypothetical primitive protocell. And I thought, wow, it's not so complicated. It's not like the Krebs cycle or something enormous. And that, given your interest in bacteria, the minimal organism and synthetic biology, I thought, boy, if there were something on that, it might be fun. But I didn't mean to push you in that direction, but I thought it would be worth at least taking a quick look at.
No, I mean, I've looked at the same Wikipedia article and I thought it was interesting. I just couldn't, like, there's a lot of people saying stuff about it, and I just couldn't find like definitive this process.
Right.
I'm sure it's out there somewhere, maybe, but I know it. But so instead, I stuck with kind of the TNC stuff and like incorporating that into a bot network. And I found this paper. Might have forgot the link on Factual on that, but it was incorporating TNC data into like genome scale metabolic network reconstructions, and I thought it would be like a good semester project to like kind of recreate what they did a little bit, and then also use it to generate data for maybe some sort of surrogate thing in the future, just like learning how the whole process works. But
So-
essentially what they did is they used the TN-seq data to like build core models of, I forget the organism they use, but essentially like smaller versions of the actual like starting network and model that they used. And then they did something kind of similar to the idea I had of like masking based off that data. That's not exactly what they did. I'd have to go back through and like figure it out, but they essentially used that and got a bunch of different like core models that like theoretically could work or whatever. And so I thought it would be interesting to like recreate what they did, get all those models, and then use them to like generate some data set solving, some just flux analysis, and then kind of just go through that whole pipeline for, I don't know, I'm intrigued with the surrogate idea, and I still don't know what it looks like, but like getting kind of the starting point of that, like how to incorporate kind of weird data to like try and figure out a little bit more of how do we map out the.
Yeah, I mean, I think it's a fascinating problem. It's pretty far from my expertise, so I'll have to learn along with you on that one because
I think luckily, like they have the paper, they have everything there, so I can like kind of follow along and have a couple steps afterwards. So, but
this certainly has a networks component. It also has a more classical bioinformatics component to it, and so I'm less familiar with the bioinformatics approaches. The I mean, I was working Monday. I was meeting with a grad student that I'm helping advise, and who's in the Netherlands, and he's been working on these very elaborate multi-scale models of early embryonic development, and they're quite sophisticated, and they have relatively detailed molecular networks of regulation and signaling inside of the cells of the model. And something that I had not really heard him talk about much, which is new work that he'd been doing, is he'd been doing a bunch of experimental work on gene expression in he's working in fish, in zebrafish. And then he was using a protein interaction database and an LLM that was then predicting from that protein interaction database effects on particular pathways which existed in his computer simulation, and then looking at the computer simulation then to predict actual macroscopic phenotypic outcomes. And he was actually able to make predictions and validate them experimentally, which to me was pretty remarkable. We talk a lot about genotype to phenotype or molecule to phenotype prediction. And that's been a dream, but it's not something that people are very easily able to do. Now, in your case, when you're focusing on metabolism, you're sticking inside single cells. There's nothing wrong with that, but it impressed me. Surprised maybe is the wrong word because he's a remarkable student, but it impressed me that he was able to get essentially from give it a molecular species and predict macroscopic phenotypic output at the tissue level of this organism. And it did have this kind of inference network as a big part of what it was happening. But again, it was leveraging rather elaborate multi-scale model that he's been building and validating now for several years. So, but the basic principle of the kind of workflow you're enunciating is similar, and so
yeah, that's really interesting. I just don't understand yet how to incorporate like the dynamic aspect of with
well,
some of the more FBA stuff.
Well, I mean, it may be that if the point of the class is that you learn something and that you enjoy yourself, and so if it winds up that the kind of networks you're using are not dynamic ones, well, then the overlap with the other things you do in the class may be less, but that's all right. I'm not gonna be insist too hard on that. He gave a virtual seminar on a lot of that work this earlier in the year, so I could also share you him giving a talk about that work as well.
So I have gone through all the applications that I've mentioned in the previous class. So this seems like something that I can put together right now with the literature study I did, and then not complicating the model like more. So what I have chosen here is cell proliferation and how the cell proliferation is behaving with respect to glucose availability. And I have chosen two particular glucose inhibitors, which is GLUT inhibitor and hexokinase inhibitor. And so I also managed to get a mathematical model which is designed for all these parameters. So this is from one paper where they have traced the cell proliferation with respect to the glucose availability. And there is one more paper like where they have talked about like what is the rate based on these inhibitors with the cells with the glucose availability. So I thought like I can combine these two models and I can track these objective statements, like how the simulation of the cell proliferation goes through and how it varies with respect to the inhibitors, that two inhibitors which I have chosen. And I can also go through all the parameters of the rate constants to see which affects which and then analyze the complete process. So, um, uh, but then the models that I have here, all are MATLAB versions, so we have to convert it to Antimony if we are going to use that.
I think that seems like a practical thing to do in a semester if it's something that is of interest to you. I mean, one thing you might want to think about, and I'm not sure how practical it is in this context, is one problem always with chemotherapeutic approaches is that cancer cells are very rapid evolvers. And so almost any chemotherapeutic you apply, the cancer cells evolve around whatever the pathway that you've been affecting is. Now, in this case, since you're affecting nutrient availability in a generic way, maybe it's harder for the cells to evolve around that, but I suppose they could modify this as the drug is affecting a transporter in this case. Is that
yeah.
No. So I suppose in principle that there could be some kind of mutation in the transporter that reduced the efficacy of the drug. I'm not saying you have to model that, but whenever I think of cancer modeling of evolutionary effects in cancer is always something that comes to my mind.
Yeah, so in the cancer simulation course here with Loveless, we built this cancer model considering evolution, but we already have we already know what the evolution looks like.
Mhm.
So we have two populations, for example, one population of cells that consume glucose and another population of cells that consume something else. Because since there's no glucose availability, they start consuming something else. And it's I don't remember what exactly that is. I think it's they start consuming some byproducts that are very available. And what we do is we model a flow from one population to the other. And this flow depends on the glucose availability.
Mhm.
So this is something you can do, which follows more or less what James is saying. So yeah, you can definitely start with that.
So that's actually an interesting control problem.
Mhm.
So if you take, I mean, that we were thinking of trying to think about ways of building a control problem. So that's actually, I mean, it might or might not work because it may be a little too simple. But suppose that you have, as he says, two predefined cell populations, which differ in some set of parameters. It's difficult to do pure evolution in these kinds of models. You could do parametric evolution, but fundamentally you only have one replicate of the model or two replicates of the model. So you have two networks which represent the same network architecture, but different parameter values, which would represent some mutation in the state of the cell. And now you have a global model that represents the replication and population of cells of those two types. And so now you compete them in the way that Pedro said. And now you suppose if you simply give the chemotherapeutic on some schedule, then you will select for whichever cell type is more resistant to that chemotherapeutic. And so what you'll see is that the population of the resistant cell type goes up versus the population of the less resistant cell type. Now, the net result may still very well be that the tumor grows more slowly than it would have without the chemotherapeutic, but it will eventually escape the chemotherapy. You could then ask the question, for example, suppose that I give the chemotherapeutic in what's called a metronomic fashion, where you give the chemotherapeutic and then you have some interval where you don't give it. When you give the chemotherapeutic, you reduce the cell population, you favor the resistant cell type. During the interval when you don't give it, the non-resistant cell type, if it has a higher intrinsic proliferation rate, will then win out. And so by giving the chemotherapeutic at particular intervals, it's sometimes possible you don't eliminate the tumor, but you can control the size of the tumor to keep the maximum size of the tumor limited at the same time that you don't allow the resistant type to take over. And so one would have to think a little bit more about how to do it, but in principle, you could do these kinds of competition experiments do allow you to look at optimizing dosing schedules. Or if you have, for example, as you point out, potentially two different chemotherapeutics, which ideally one of the cell types is susceptible to one, the other one is susceptible to the other, then by changing the pattern of dosing or doing simultaneous dosing, you can then get better outcomes. Now, that's still pretty vague. One would have to do some real work to try to make that more specific, but I think that that's a potentially quite interesting. The problem you have here is already fine. I'm not saying that you have to do the evolutionary or personalization optimization, but since you started out by saying that therapy optimization was something of interest to you, I'm trying to think a little bit, brainstorm a little bit about ways one might take this idea and try to take it in the direction of personalization or therapy optimization. Great. All right.
Can you share me if there is any reading material regarding evolutionary modeling?
Maybe I still have the model somewhere, I can look for it.
Okay.
If not, we can just discuss about it.
Yeah.
Yeah. I
Well,
mean-
what you have is, as James says, is enough for a project.
Mhm.
But if you're interested in like, treatment with cancer evolution, with as few as two cell populations, so, there you show just one, you have N. You could have NA and NB for two cancer populations. And then you can, have a simple evolution model.
Okay.
So we can, yeah, there are tons of things you can do, and I can definitely help you. I'll try to find the model I built in class. If I don't find it, we can.
Thank you.
I mean, essentially, the problem always with simulating evolution is that at some point you have to define a trade-off function. Right, the more resistant cell type has to be less, has to have a lower growth rate. If the more resistant cell type intrinsically has a higher growth rate, then it wins. There's nothing to play with.
Yeah, that's.
And when you pick the trade-off function, sometimes you sort of game the system in a way where you know what the outcome is going to be beforehand. On the other hand, if you're allowing things like different dosing strategies, then even the game system can give interesting results.
Yeah, for glucose pathway evasion, that's very typical. Cancer cell that finds a replacement for glucose usually grows much slower because these other calorie sources less dense, less efficient. So yeah, usually there is a trade-off.
Okay. So I also had one more idea
Yep,
where
okay, go ahead.
I got to look at the drug combinations, and it had like multiple targets.
Mhm.
But the factor is like how complicated I may wanna build this particular system so that I can test them for all those targets or like more than two targets, because here like I'm just tracing the glucose response towards
Right.
the cell. But then if I want to include multiple targets, like how do I put through it and how complicated the system gets?
Target 4.
Multiple inhibitors,
Right.
not only glucose.
So you've just addressed something that's very fundamental, and it's an intrinsic limitation of modeling, which is if you haven't included a pathway in your model, you can't simulate what happens when you perturb that pathway. And so there are a lot of people who do modeling who believe in what are called minimal models, where you build the simplest model that's capable of explaining the data sets that you have. And those are often very, very effective, but they're limited in the sense that if you then want to extend to consider other things, it's hard to do that. And so, for example, if you wanted to model, classic cytotoxic chemotherapeutics maybe aren't that, it's a little bit less of an issue. Although even for those, there are ways that cells can avoid them, evolve around them. But for example, for things like targeted chemotherapies, where you interfere with particular signaling pathways, then if you don't have some representation, gee, this cancer is growth factor dependent. If you don't have the growth rate regulated by the pathway related to that growth factor, then you can't really simulate the effect of a treatment on that. And so you would have to look for slightly more complex cancer models, and they can get very complex indeed. I mean, maybe too complex, but it might be worth doing a little bit of literature review of models that maybe have some additional pathways in them, which then you could target with a particular therapy. On the other hand, getting something like this under your belt would be good to start, because this is something which is clearly achievable, you can get it done.
Mhm.
And while you're doing that, you could look for slightly more elaborate models. And then if you find them and you're able to do it in the semester, that's great. But if you're, again, my goal is to make sure everybody has success. And so, but I'd be happy to sit with you and do a little bit of trawling of the literature to look for models that have a little bit more detail in them.
Okay.
For potential drug targets. The example I mentioned of the student in the Netherlands that I've been working with, he has pretty elaborate gene regulation models in his cells. And so that gives him plenty of places to look to target. If you don't have that level of complexity in terms of gene regulation, then you have less ability to simulate the effect of interventions. And so that's a reasonable thing to ask.
I do find something that is more specific than last time. So I said that I may choose like one or maybe two. A few signaling pathways. So I did some search and found this paper that has a signaling pathway that has parameters. It has equation like rate equation and lot of rate equation and also the meaning of each parameters and also the value of each parameter. So I can reproduce this very efficiently, very quick. And they also have some initial concentration. So it's reproducible, and I may try to like use some other data set to like test if adding some drug, how will it affect the signaling pathway. And it has some, because like some drug will cause the cell to die. And I will see why it will cause the cell to die, like what protein changes and like adding it at what time frame and how it will affect the cell growth, something like that.
Right. Well, so that actually was a perfect answer to your question. So you're starting from a very high level model with a very simplified minimal model where you're focusing on glucose metabolism as the main regulator of cell proliferation and survival. And that's a very reasonable strategy. You're starting from a model of a specific pathway, the PI3 kinase pathway and the MAP kinase and its downstream effectors. And so there's a very specific model of a particular set of pathways. And so in one case, you have the ability then to ask questions about perturbation of those particular pathways and drug targets. On the other hand, those models may not include the glucose transporter that you were looking at. And so in a sense, you're taking a bit of a bottom-up approach, and you're taking a more of a top-down approach to addressing similar questions. And so I think, I'm not saying you have to work together, but I think it would be interesting for you to talk to each other and to see perhaps there are some models that incorporate both perspectives. They may be too complex, in which case it's okay. But in the one case, you're starting from a generic, essentially an external observation about how the cell behaves under particular nutrient conditions. In the other case, you're looking for starting from a particular mapped pathway that you know is active in the cells that's relevant to cell proliferation and survival. Those are both valid strategies. In some ideal world in the future, you could imagine those all being harmonized in some meta model that includes both. But to begin with, they're somewhat different perspectives. But I'm really interested in seeing how you're thinking about these problems. And having some existing model to start with, I think, is always a good idea. It's Now, occasionally, I'll warn you, and I don't think it's the case with any of these. Occasionally over the years, somebody has started with a published paper and then discovered that the paper was wrong. There are a couple of times over the years when I could think of a student said, gee, I'm really interested in this. This paper is very interesting. And then they've tried to repeat the results of the paper. You'd say, well, it's a simulation paper. How could it be wrong? The answer is somebody made a mistake. And so there are a couple of times when actually people have started with published papers and the referees didn't catch that there was a big mistake. I don't think that's going to be the case of these, but it has happened. So just because it's published does not mean it's right. Okay. All right. So let's move on. I want to come back to these concept that I talked about last week, which was this idea of how do you interpret a diagram into a set of mathematics that can be simulated, and the idea of the use of a Hill function as a way of doing that. Here we started out with the idea that P1 activates P2 or P2 inhibits P3. We're thinking about P1 and P2 as transcription factors or activators or inhibitors of transcription. And the reason I was reading Wikipedia about epigenetics was because I was thinking we have these incredibly oversimplified ideas about how gene regulation works. And back when I was a student, our understanding of epigenetics was really very limited. And so I thought I better just do a little bit of a sweep to see what's happened since I last thought about it. And the answer is that there's an incredible amount of detail in terms of epigenetic regulation of transcription, none of which is ever included in these simulations that we do. And somehow the simulations still seem to work pretty well. But the basic idea here would be that you have this concept that P1 turns on the transcription of gene 1 that then produces messenger RNA that's then translated into protein P2. And we write that in a very simple way as nothing goes to the protein because we don't actually model the components that produce the messenger RNA or the protein. And we embody the production rate of the protein as a Hill function of the amount of the promoter, P1, or the. Similarly, if we have an inhibitor, we would model this as nothing goes to the protein P3, and we have a sub-basal rate of transcription plus some decreasing Hill function of the amount of the inhibitor. Now, in reality, for the most part, there is some regulation of the rate of transcription, but in general, you're either transcribing a gene or you aren't. And so the production of the messenger RNA tends to be in bursts. Those things tend to get smoothed out when you get to the translation phase of the protein production. And just reminding you what Hill functions look like, and also the fact that here at the top, that when you're going to model protein production, when you see a lollipop arrow like that, you always have to remember that you have some kind of degradation of the protein. Otherwise, you'll wind up with an infinite amount of it. There's some detail here, which I have left out of our detailed discussion. There are different ways that you could parameterize these Hill functions, and there are ways of doing it that make it more convenient to do things like ask the question, if I change the maximal rate of production, what happens to the amount of protein? And there's some scaling arguments, which in some years I've gone into in detail, but in general, yeah, go ahead.
On the homework, you had like examples of how to go from the picture essentially to like the
Right.
equations.
Right.
And I was sort of initially confused on a few of them because they weren't just the generic, like either you have A on top for activation or you have K on top for repression. And is it pretty much just on top? If you have activation, you want it to be any species that like, if it increase, like any concentration based, I can pull up the exact question I had, but like,
So I think I want to hear your question, but I have a feeling I know what you're going to be asking. Which is, suppose that I had P1 and P3, both of which affected the production of P2.
Yeah.
And so you have multiple signals coming in for the same gene. And that's, I won't say always the case, but it's actually usually the case. And in several of the circuits that we had, we would have either two activators coming in to the same gene or an activator and an inhibitor coming into the same gene. And you have to ask, how do those signals interact with each other? And in general, from a biological point of view, that's a pretty complicated question. Mathematically, there tends to be, and that's why there's this little logic diagram caricature at the bottom for gene regulatory network, at least, which is if the concentration of P1 is less than K1, the gene is turned off. If the concentration of P1 is bigger than K1, the gene is turned on. And that's how a Boolean network approach would work. And in those cases, you actually are essentially, in the Boolean case, you're essentially stuck. The only things you can do with that are either an and or an or. You either say, if you have activator 1 or activator 2, you turn the gene on, or you say if you have activator 1 and activator 2, you turn the gene on. For a Boolean network, that's what your choices are. There's a general, and I actually have again a whole lecture which I've skipped, which is nothing but discussion of possible strategies for doing that. Herbert has a different textbook in which he goes into some of the many possibilities for this. The general rule of thumb when you're doing these things is to say that if you have what are called coherent signals, that is P1 and P3 are both activators acting on P2, or P1 and P3 are both inhibitors acting on P2, then you OR them. And if you have incoherent signals, P1 activates and P2 inhibits, then you AND them. The general belief is that inhibitors usually are stronger than activators, so that if you have an inhibitor fighting an activator, the inhibitor tends to win out. However, Herbert, in some of his examples, and certainly if you look in the literature, there can be some pretty complicated, essentially experimentally measured cases where somebody actually measured the rate of production of P2 as a function of the concentration of activator and inhibitor, and then phenomenologically came up with a description of it. And so for the purposes of today, we're going to use that kind of and or logic. And that was what was used primarily in the homework, in the written out equations for the homework. But it's important to remember that that's a first guess about how to do it. If you don't have any experimental information, the best you could do is guess. And those are the guesses people start with. But then when you start validating against experiment, you often find that the interactions are much more complex. That's why these sort of logic diagrams of gene regulatory networks that make them look like integrated circuits, are a bit misleading. Because when you see those diagrams, you say, well, it looks as if there are 8 signals coming in to control this gene. It looks like I ought to be able to just say A and B or C and D, not E. Well, maybe. And that, as I mentioned, it neglects entirely the epigenetic regulation of the transcription of the gene. So again, for our purposes here, we're going to assume that things are simple, but it's important to remember that they may not be. I was going to do a little exercise here, and then I decided not to do it. I just want to remind you about this variable stoichiometry. And we're not really going to use it very much in the class, but it is allowed to have the stoichiometric coefficient here be a variable instead of a number. But tellurium and antimony have some rather complex and finicky rules about this. I can't say E plus NS goes to ES and then ES goes back to E plus NS, which would be the natural thing to do. That whatever that variable name is can't be repeated between equations. And also, if you try to change the values of N and M from Python, you have to do it indirectly. And I'm going to talk in a second about this colon equals notation, which you see here for what are called functional relationships. And then the reason I dropped this as an example was I was going to have us calculate the Hill functions for different exponents. But to get the Hill function, this calculates the time series for different exponents, and that works. But the natural way to calculate the Hill function is what we did last week, which would be to put a $sign in front of the S to freeze the concentration of substrate. And it winds up that J2 ES goes to E plus M times $S will crash. You're not allowed to fix the concentration of the target species when you have variable stoichiometry. And so you can't actually do this calculation the way I had planned on doing it. And so I'm just going to let you know that it's possible to change the stoichiometry if you want to, but it's a little bit finicky. Leave it at that. So the next thing I want to do is more fun, which is to talk about downloading pre-existing models and converting them to antimony. There are a number of model specification languages besides antimony. I like antimony because it's easy to read and write, but the 2 standard model specification formats that are used for interchange are something called system biology markup language, and then also CellML. CellML maybe isn't used quite as much these days as it used to be. It's used more, it was always used more in the pharmaco-kinetic world than in the molecular signaling world. CellML came out of Auckland, New Zealand, Peter Hunter's group. SBML was largely Herbert Sauer's initiative. Both of them have large numbers of models. And these are what are called exchange languages. They're both markup languages. They are sort of human readable, but in general, you don't want to be reading them by hand. You normally use some kind of editor that interprets them for you. We can download models written in SBML and run them directly in Tellurium without translating them. And antimony and Tellurium also provides services to translate those SBML models into antimony, in which case they look like regular text and they're easier to edit than they would be in SBML form. You lose some details. SBML allows you to do things like specify the layout of the network and a variety of other graphical information that you lose when you go to antimony. And so I'd like to do this together. We're going to use the biomodels repository, which is a British model repository. We'll download the model, we'll upload it to Colab, we'll open it in Tellurium, convert it to antimony, run it, and then we can save it as an antimony model. And so I'd like people to do that. So step one, I'd like everybody to go to biomodels. And if you search biomodels, you'll find it. I'll give people a second to do that. And I don't know if anybody's tried biomodels already, but if you haven't, it's an impressive repository. It has thousands of validated models in it. The next thing that we were going to do was to pick a model, and my suggestion was EGFR, although you could pick your favorite pathway or topic and hit search. And it should pull up a list of possible models related to that topic. And in the case of EGFR, the one that I picked was the Bianconi 2012 EGFR and IGF-1R pathway in lung cancer. But you're free to pick another model. Not every model that they have is actually executable. And so I picked one which I know works. So everybody got that one? Yes.
On the first page, they have this browse by modeling approach. Is the converting something into antimony only work for like protein models or?
So browse by modeling
Please
approach.
scroll down.
You can find models here that are not instantiable, well, not executable, where you have the pathway architecture, but nothing that would give you the information about how to actually make a dynamic model.
But these are all dynamic.
No. There are models in biomodels that are only the model structure.
And by that, you just mean like the prediction.
Yeah. No. And so I mean, the reason I picked the example of Bianconi was because I know it would work. Probably 2/3 of the models that are in the biomodels will be fine, but not everyone is. So for example, there's a subcategory of SBML called SBML qual for qualitative, which is only the model structure without the parameters or rate laws or anything else. Just says this activates this inhibits this becomes this, but it doesn't say how fast. So models like that, you'd have to do the kind of thing that I was showing you earlier to guess what the rate laws were to make that executable. So for the moment, I'd ask your indulgence and pick that Bianconi 2012 one. You're going to get in a few minutes the opportunity to go and try any one, but I thought we'd start with one where we know it works. Does everybody find Bianconi 2012? Yeah, well, let me walk you through the steps, and then we can come back. Leave your thing loading, and I'll walk you through what you have to do, and then at least you'll know what to do once it loads. Because I'm a little puzzled by that, because as I say, when I launched it just now, it loaded instantly. So you're going to pick a model. As I say, I suggested Bianconi 2012. And then when you double click on it, it will open up a description of the model, which will have a session number. So there's a unique identifier for the model. And then a description of what the model is, and the abstract from the paper, its publication history. And it will also have a related publication link, which should take you to the original paper. And down at the bottom right, there's a little graphic And if you click on that, it should display the network that's being modeled. Now that network editor that they use to do that display is not really very pretty, so it's not something that you'd want to use for publication or anything like that, but it does let you actually see the architecture of the network that you're going to play with. And let me actually see if I can get that to work for you, since people are having an issue with it. Okay, so I'm going to look for EGFR. If this decides not to really, if their website is really down, then we may have to jump ahead to something else and come back and see what's going on. Okay. Well, I'll turn the screen share over to you for the moment. Okay, so that's the network. As I mentioned, those networks are not very pretty, but you can at least get an idea of what's in the paper from looking at them. And now if you can go back to there, if you go up to the top, there should be files, go under files, and you should be able to download the model. And the model is that top one where it says download. There you go. Download that. And now if you open that model in a text editor, you can see what it looks like. Let's go back to the page with the model, with the list of models. That should be the XML. That should be the actual code. There we go. There we go. That's it. So this is what SBML looks like. But wait a second, hold on a second. Only part of the file is loaded here.
I think that's
Keep. But this is only the header. It's not the actual model. Can you scroll down? Oh, there it is. Let's see if, yeah, so there it is. So this is actually the contents of the model. And as I mentioned, this is not the most enjoyable thing to read for a human being. So what we can do now, and I'm going to borrow the screen share back for a sec. Well, actually, you can leave the screen share. Well, what I want to do now is walk you back through the next steps. So the next thing that one's going to do, you find the model. As I mentioned, you visualize it, which you saw. You can see the original paper, which you saw. And then we're going to do a download. And that first file that it offers you, where it says SBML level 2v4, is the one you want. So you hit download. And now in your Colab, what you want to do is you want to load SBML model and then the name of that file. And so that's what we want to do. So if you open Colab, you're going to have to do your usual imports for Colab. Import Tellurium is TE, and now R equals TE.load SBML model, and then a pointer to your file. So does everybody now have that biomd 000000427_URL_.xml? And so now launch Colab, install Tellurium, and then use the command R equals TE.load SBML model. And that may actually be a little bit of an issue with Colab, because you're going to have a pointer to the model. If it is an issue, you can literally cut and paste the instead of TE load model, this line here, TE.load SBML model, You could do triple quotes and then paste the whole thing in. And then triple quotes. That'll work. So I'll give people a minute. Was everybody able to get it to load? The issue with Colab is that you have to figure out what directory you're pointing at. So that could be a bit more of an issue. Looking at it now, I realize that this slide dates from the time when everybody ran Antimony local, so I didn't have to explain how to point to a file from Colab. And then what's interesting is there's this command r.get antimony that will convert that SBML model into an antimony model, and then you can do a print antimony model, and you'll see the converted versions. And then you can create a cell called antimony model equals triple quotes, paste that in, and now you have a string which contains the antimony model. So what you're saying is if you want to execute it, do I need to paste in the string for the antimony?
Yeah, like, can you just do te.load a,
And then I could do r.simulate will work.
oh, wow.
So I can simulate the SBML model without translating it into antimony.
Oh,
So I can do r equals te load SBML model and then do simulate, and it will run. So you don't need to do the translation. This is showing you that if the model is only available as SBML and you want to turn it into antimony, how to do that. Again, the goal here is in terms of execution, SBML is what's called an exchange language. I mean, nobody reads PDF, or in the old days, nobody read PostScript. Those were exchange formats that computers used to communicate with each other, but you weren't meant to read them as a human being. I mean, Markdown in general is not something one likes to read very much. And so the goal here is just to show you that even if the model exists in a format that you don't like to read, like CellML or something else, you can pull it into antimony and then have something that's easier to edit. So now you can do that r.simulate. And as you point out, I don't need to have done that translation to do r.simulate. That r.simulate's actually simulating the SBML. And then I can do an r.plot. There's also a tool online, and this may or may not work. If biomodels is being difficult, this may not work. If you look at sys-bio.github.io/make-sbml, this is a web-based interface that allows you to translate models and even at some level execute them online. So can people find this? It's sys-bio.github.io make SBML. See if you can find that. And let's see how it's working today. So if you now go into the search biomodels, we can type, let's try EGFR. It has that Bianconi model. I'm just trying it for myself first before I do it in public to see if it works. All right. And when I load that model, it actually will automatically do the translation. Did that work for you? And actually it came up right away. I know historically when the biomodels repository has been slow, sometimes this website would fail because of timeouts. So here we are in the make SBML model. So I started out with here, I'm going to search let's see. Cancer metabolism. No guarantee. I don't know whether these models are extensible or not, but let's pick one. Pick that model. Here's the XML, the SBML model. And here it's translated into antimony. And this one looks pretty complex network architecture, and it has parameters. So this is actually an executable model. And so I could save this model here if I want to. And I could also go the other way. In other words, if I had an antimony model and I wanted to turn it into SBML, I can put that, either upload that antimony model here or paste it in by hand and then hit convert and it'll turn it into SBML. I can do that with the commands. I could do that with the line commands in Python. But if I don't have to remember how to do it, or I don't want to have to launch Colab or something else, I can do it at this website as well. And the ability to search biomodels automatically is pretty nice. Okay. So my little exercise then was for people to go to biomodels and sorry, got the screen share. My little exercise now is to go to biomodels and find a model that's interesting to you. Find a model that's interesting to you, and see what, and download it and run it. So if you're interested in cancer, you might pick some kind of cancer therapy model. If you're interested in metabolism, you might pick a metabolism model. See if you can get it to work. You can either use that get antimony command, or you can use that website that we just looked at. And you can tell me what model you picked and what motivated you to pick it, and did it actually run? And if it ran, what did you get? We can have people screen share if you like. As I mentioned, most of the models in biomodels are executable, but not all of them. So you can be unlucky. That's one reason that it's worth looking. When you click on the model and you read the model description, it will tell you in that header page whether the model is executable or not. And I'm going to try this myself while we're at it. I picked a microbial metabolism model myself. Here, I'm going to grab that. You want to screen share your example? Let's take a look. No, that's certainly not doing anything. Let's see. Let's take a look at the so this has got lots of chemical reactions. What was the model that you picked, by the way?
Colab metabolic depth.
Adaptation, okay. So you've got a bunch of rules there. That looks okay. Let's look at what happens to the parameter values. It has parameters. So sometimes one does have to actually read the paper and see, because the default parameters may be for some situation in which nothing is happening. So why don't we go from 0 to 1, just time 1. See if that helps us. No, it looks like nothing is changing. That's a little bit surprising. Not crashing on you, so I think they're so we would then have to go back and look at the paper. Okay, first tell us what model it was, and then
So I picked insulin glucose dynamics.
Mhm.
I did not see a model structure nowhere, but there is a paper. And
if you scroll all the way down to the right, so go back to the biomodels. Yeah, no, they don't have a layout, do they, for that one?
yeah, I was looking for a diagram somewhere.
No, it's probably an old enough model that it doesn't have the layout embedded in it.
Yeah.
Tellurium will actually has a facility that will generate layouts of the network, although it's not generally very pretty. Okay, but there's the paper. Okay.
Yeah, yeah, this is the workflow, not the model itself. However, they do have the equations.
Yeah.
Which are okay, but these are just a few, they are not all the equations.
Right.
It does run, so I have it here. It's this huge file. It doesn't really tell you what each thing is. So R0, R1, R2, like the names don't actually mean anything. Well, I guess mTOR does blue.
PI3 kinase, yeah.
Yeah, but even then I would have to go back to the paper and read through it to understand what's going on here. Because there are so many equations. And it does run. So like it seems that 99% of their species are actually not doing much. They have 3 that are doing something and the others stay at 0. So maybe they do play a role in other tasks like ingesting something, eating, exercising, or diseases, which is the focus of the paper is diabetes. So it runs, but to do something useful with it, I would have to really understand what it did.
Another possibility, Pedro, in that case, is that they've scaled the values of the variables differently.
Yeah.
And so you notice that some of your things are going up to values of 40,000.
Yeah.
Some of them may be scaled between 0 and 1. So it's possible that if you select some columns, that they may not be so trivial.
Yeah, if I just do R2, we have something.
Yeah, see, those are scaled between 0 and 1.
Yeah. Yeah, definitely, that's good catch. Yeah, so these may be playing a big role, but I didn't see the previous one.
Okay. Ellen, were you able to find a model to download? Okay. Okay, how about you? You said you were able to find one? Okay. Well, let's take a look and see whether we can. I hadn't really planned on spending the class doing this, but it's fine. It's important to be able to get this to work. It's once you've got it working, it's a big resource. So so there's a so it looks like you've got a model that's not instantiable. All right. So it's giving you an error, unable to read SBML string due to parsing errors. So let's try going to that website that was doing the translation, the automatic translation website. That one. Try loading it there. Is that where you got it, or is that? Okay, so why don't we try to find go to the auto translation website and now search for that model. It's a COVID model. Okay. I actually know the author pretty well. I haven't seen her in a long time, but we organized a big semester conference workshop together many years ago. She's at UCLA. Andrea Bertozzi. Okay. And so it looks like, okay, hold on. Don't go back and do it again because it's throwing an error there. Just put that. Don't hit OK. Okay. So, but this is telling you, scroll down. What this is telling you is that as far as SBML is concerned, there's a syntax error in that model. So, I mean, these days with an LLM, you might be able to debug that model, but I would recommend for today, pick a different model. I mean, I'm happy that it's actually giving you an error rather than just than running junk. But can you find another? Possibly search for another model. You could do it in here. Okay, or either way. Is there another one that you think is interesting? It might be. You don't have to look for EGFR. You could pick any topic you like. Okay. All right.
But
Yes.
that morning actually.
I'm sorry, I didn't hear you.
So here, the same one gave the header, it actually followed some.
So when you ran it directly as SBML, it ran. So how did you load the model there?
So after loading, when I'm trying to get a model,
right?
but then the model I run moving.
Okay, so that's interesting. So you're actually running the SBML model. And so the SBML model is running correctly. It's an SIR model of some kind. But there is some issue with the SBML where it's not translating to antimony. And so that's not impossible. In other words, the translators are good, but they're not perfect. And so the what that's telling you is that the SBML model is in fact correct, but there is something in it that is not compatible with the antimony syntax. Now these days, perhaps one could try with an LLM. You could say, "Hey, Claude, can you translate this SBML model into antimony?" and see whether it does better, or you could try the antimony model and try to fix it. But one of the problems with SBML is that because it's hard to read, if there's a parse error like that, trying to find the parse error is not a lot of fun. And so I'm not I wish I had an answer for you on that one. When I've done this exercise in the past, most of the time, most people's models work if you pick one at random, but that does happen from time to time. Okay, how about you? Did you find something? What's wrong? Well, why don't you go to the website the auto translate website that I put in the chat. Okay. Why don't you screen share and then go to the auto translate I put in the chat, the auto translate site. There you go. Good. Okay, so now that you're there, why don't you up at the top where it says search biomodels, up there. Okay. Go into that search box where it says search biomodels. What would be a topic that you would be interested in? All right, well, there are lots of cancer models. Okay, so hit cancer, and now why don't you pick, you have AKT signaling, you have carbon metabolism, you have, let's not do the Bianconi one because that's the one we just did. But I don't have any particular, okay, that one would be fine. That's a drug resistance model. All right. Let's scroll down on the left-hand side. Let's scroll down and just make sure there are parameter values for it. Species initializations. It looks like there are. Let's try it. So now try save antimony to file would be all right. And you could also copy it to clipboard. Copy antimony to clipboard. There we go. And now if you have your, if you open Colab, okay, and now in the first one you want, you'll run that, okay. And now jump down to the third cell where it says TE load quote, and then paste, keep the TE load A, but just where it says your antimony model here, paste it in. Okay, good. you might want to hit save. You better save a local copy because this is the master copy. So you'll have to go under File, at the top left where it says File. You'll have to do save a copy. No, under File at the top. In the pull down menus at the top. Okay, copy to drive will work. Okay. And now you'll have to create a cell below the cell where you loaded the model, where you do r.simulate and r.plot. We don't know how long we'll have to do it for, but we could try 0 to 10 and you'll see what happens. Okay, and then r.plot. And take another few minutes. Okay. All right. And now let's see if it loads. Okay, so it ran. It's a very complicated model. It's got a lot of species. And the problem is one of those species has a value that's numerically very high compared to the other ones. So to actually be able to explore this, you'd want to pick out the species you want to plot, adjust the vertical scales. So let's just do one thing that'll make life a little bit easier. If you go into where you did your r.simulate, say result equals r.simulate. Okay, and then use te.plot array on result. Plot array, okay, on result. Now run that. It's not going to change anything. Okay, and now we had to do a reset beforehand. So we better okay, that'll work too. Okay, and now let us adjust the vertical axis. So inside of the t-plot array, you can do y-lim. Y-lim, Y-L-I-M. And then you can set the lower value and the upper value. So 0 to 500, for example. But you're going to have to do, instead of, well, you can reload it. I was going to say you could just do a reset before there. Okay, so now you can actually see some of the behavior of the model. Now, if you ask me what that model is doing, I can't tell you because I haven't read the paper, but at least you're able to pull in and run a fairly complicated model. And the whole point of this exercise is not that it gets you off the hook from reading the paper and understanding what the model does, but the goal is that you can actually replicate these results. You don't have to compile a bunch of C++ code. You don't have to use any commercial software. You're able to actually share models relatively easily. Great. Okay, so that worked for you. Ellen, what did you find? You can screen share if you want. Sorry? Well, I'd prefer it if you didn't simply take a model that existed and give me the model exactly as it was there. But if you want to start with a model of that kind and extend it or combine models or do different analysis, that's fine. Right. Right. So the whole point of these kinds of model sharing sites is that the goal is for you to be able to adapt and reuse models. And so if, for example, there were a model of cancer cell growth that you were interested in, and you wanted to take that and put it into a machine learning framework for doing control of the cancer, that would be fine. Or you said, gee, there's a model of cancer cell growth, but it's missing this pathway, and there's this other pathway model, and I want to put the two things together. That would be fine too. So one thing that you will find is that even if there are two models that have the same chemical species in them, getting them to work together is not trivial. It's not that you have things that you can just plug together automatically. Okay, so tell me, spell it for me, please. Jared, here, this one. All right. Here it is. All right. Somebody's annotated it really well. It's got all of these things where it says identifiers. Those are ontological annotations. I'm sorry, the way the screen is set up, if I make it big enough to read, then I don't get the whole field of view. So I'm gonna do this as copy Antimony to clipboard. And then I'm gonna go here. And I had a model load here. You just put this model in here. That. Make sure I'm screen sharing it all right. I put it in there, and now I don't need this fancy thing here. I just need to r.simulate. So we have comma 10,000 r.plot. See if it works. Oh, I left out my parentheses. Okay, so that model works. I don't know what it does, but it works. Time scale is not right. So it's synthesizing something. I don't know what it's telling us, but it does actually run. So the model is downloadable and executable. Okay, thank you. I recognize that if you download and execute a model without knowing what it does, then the likelihood of getting something meaningful out of it is moderate, but At least we have the basics of how to download and run them. Well, I guess we're not going to wind up getting to the toggle today, but we can do some other things. All right, so I wanted to talk a little bit about some very simple antimony code. You've actually seen this antimony code already in the simulations that you've been doing. So why don't we just, we're going to be talking about functional relationships and also ODE declarations, both of which you've actually seen in the simulations that we've just run. But I want to talk a little bit about the syntax. So why don't you clear out, you can use the same notebook that you've been using, so you don't have to reload Tellurium and wait for it. And write a very simple simulation like A goes to B at rate K1 times A, B goes to nothing at rate K2 times B. Start out with some A, and here I gave K1 equal to 0.2, K2 equal to 1. And then I just did a R dot simulate from 0 to 20 and an R dot plot. And we could certainly do exactly the same thing in a fancier situation, but I just want a very simple case where we can understand what's happening. And as I mentioned, we've already seen examples of what we call functional relationships and also of explicit ODEs, but we haven't played with them in detail, and I'd like to make sure that we're familiar with the syntax and the functions. So the next thing we're going to do is we're going to modulate the value of a parameter, K1. And so I've mentioned before that the antimony string is what's called a declarative specification. And when we load the antimony string, assignments like A equals 10, B equals 1, K1 equals 0.2 are read and compiled. And that means that if we try to write something like K1 equals 0.2 plus sine of time, that will be evaluated for the value time equals 0, and it will give me the value of 0.2. And so when I run it, K1 will not change. And so if I want a parameter value to update in time, I have a special kind of equal sign, which is the colon equals. And that colon equals says, check what is happening on the right-hand side of the equal sign and update it. And we're already familiar with multiple equality types, equal sign types in Python, because we have an equal sign for assignment and a different equal sign for comparing whether two things have the same value. And so in antimony, we have two kinds of equal sign, a regular equal sign that is evaluated once on load, and a functional dependence equal sign, colon equals, that is updated continually in time. And so now try typing K1 colon equals 0.2 plus sine of time instead of K1 equals 0.2 and run it and see what happens. Thank you. Do you get a different result? It's, right. Let me know when it works. Yeah. Question?
Could make a K variant.
You mean why would so I'm not proposing that this is a very biologically meaningful this is a coding syntax example, although I suppose you could be I don't know, K1 could be the illumination intensity in the room or the illumination intensity outside, and there could be some sinusoidal variation as the light and dark. It was the simplest example that I could think of where you could get a time variability without too much trouble. You could also slave K1 to a species. You could say K1 colon equals 0.2 plus A, for example. That would be allowed as well.
Can you, like in Python where you do r dot K1, can you do this? Colon equals that?
Okay, so yes, you can, and if I had known that we were going to take so long to do the translation stuff, I would have done events as the remainder of this lecture because they don't take very long. But there is a command at parenthesis colon something. And that is equivalent to an if statement. Well, it's not exactly equivalent to an if statement. It's like an if. It's the declarative equivalent of an if statement. So I could say at parenthesis time greater than 100, A equals A plus 10. And that will make A bigger by 10 at time 100. I could also, though, do fancier things like say at A less than 10, K equals minus K. And then every time A crosses that barrier, it'll flip the sign of K. And so you can actually build rather complicated logic structures that way. One of the things that you face in biology is that in general, the control logic that we have in computing, if statements and loops and so on, are not played out exactly the same way. Inside of a cell, essentially all chemical reactions happen at the same time. There are things that look like loops in the sense that you have things like the cell cycle, but those are the result of processes that are acting spontaneously. There is no loop function that is executing a for loop inside of a cell. And so it takes some thinking about how to organize the contingencies that happen inside of cellular architectures in a way that respects the biological reality, what's available to things. And so events could be used to say, well, at a certain time, I added a chemical to the dish from the outside. At time greater than 100, add chemotherapeutic value goes from 0 to 10. That would be possible. Or I took some supernatant out of the dish. That would be possible. But there could also be something where there is a switching behavior in the cell that is triggered every time something happens. And so that event syntax is very simple, but it can be quite powerful to use. You could always do that kind of definition in the Python layer, but that requires you to run the simulation, stop it, see what happened, make a decision in Python, and then run the simulation again. And so events represent a way within the intrinsic logic of the antimony model to have certain kinds of things happen. And so, unfortunately, I don't think we'll have the slides for those for today. I don't have those exercises prepped for today, but I was going to do it next week, actually. So if you're interested in it, the syntax, again, is exceedingly simple. At parenthesis, some logical expression, colon, something that happens.
They really use that now.
Right, yes, so you did that already, yes. So occasionally, and I should have said this explicitly probably in lecture 1, I will include a methodology in the homework that we haven't talked about a lot in class, so that when we come back to it in class, you're motivated, you've played with it a little bit. And so that would be an example. Yes. So when you apply a pulse, that would be exactly one way of doing it. But that would be triggered in time. But you can also trigger off of levels. You could say when the value of A is bigger than 10 or less than 5, or when A plus B is greater than 7. You have anything that is an expression that is built around variables and parameters that exist within the simulation, either species A and B, parameters K1, K2, or time, which is always available. But you don't have to trigger on time. You can trigger on other things. So, for example, you could have a situation where as the population of a cell, suppose you were modeling cellular populations, and you had something like quorum sensing, where when the density of the cells gets above some threshold, the cells change their behavior. You might have a specific molecular model of how that happens, but you might also just assert it. If number of cells divided by volume bigger than 10, then K1 equals 10 instead of 0, and you're turning on some mechanism that was turned off before. So those are possible. It's not quite the same as an if statement, because in the case of a trigger, that trigger will execute once. And again, I'm giving you now discussion of slides that I'm not showing you. The trigger will execute once whenever the state of the trigger goes from false to true. So if you say at time greater than 100, that happens exactly once when time is equal to 100. If you say at A greater than 10, if A ever goes above 10, it triggers once. But if A then goes below 10 and comes back above 10, whenever A goes from below 10 to above 10, it'll turn on. So you can get repeated triggering. All right. Good point. So to some extent, you could use functional dependence to do something similar. I could have some logic function on the right-hand side of that colon equals that mimicked the trigger. Because I could say colon equals parenthesis A greater than 10, because that's a logic statement which will evaluate to true or false. Actually, I should be careful. I think that will work. I haven't tried. I'm pretty sure. There is certainly some way to get that to happen, but I don't know whether logic, what the exact syntax for logical operators is. You have to look at it. Okay, so did you get something that looked like that? We've talked a lot, but okay. Another thing that we could do is use time in our operator. So why don't you try K1 colon equals 0.2 plus 0.1 times time. That one isn't so dramatic. The other thing is that if I can write relationships between parameters. Suppose that I want to scale a bunch of parameters by some reference. So I say K2 equals 3 times K1, K4 equals 2 times K1, K6 equals 7 times K1, and so on. That will correctly compile in the Antimony. But if I go into my Python layer and change the value of K1, it won't change the value of K2, K3, K4, and K5, because that's evaluated only on load. And so if I want to be able to do scaling of parameters and control that scaling from Python, which I often do, then I have to declare K2 colon equals 3 times K1. And then when I change the value of K1, K2 will update automatically. And so that one is actually a little bit confusing sometimes. People will expect, if you write K2 equals 3 times scale, K3 equals 4 times scale, K5 equals 10 times scale, and then I say r dot scale equals something in Python, you'd expect all of your parameter values to change according to scale. They don't. You have to use the colon equals. So that's a little bit of a trap. And it's again because we're not writing a program, we're writing a declarative specification. And so that declarative specification is only executed once on the load. And so we have to, that's something to be aware of. All right. And the last thing that I was going to show you was something again that we've used already. I've made the comment that one of the things I really like about antimony is that its fundamental concept is a chemical reaction. A goes to B, or B goes to nothing, or A goes to, or nothing goes to A. Now, of course, a declaration like B goes to nothing at rate K2 times B is equivalent to writing dB by dt equals minus K2 times B. And you could say, well, what's the difference? And the answer is that if I write all of these arrow diagram reactions, those uniquely map to a set of ODEs. However, if I give you a complex set of ODEs, there are many arrow diagrams that could have given the same ODEs. And let me give you a simple example of that. Suppose that what I care about is the number of cancer cells versus time. The rate of change of the number of cancer cells will be the rate of creation of the cancer cells minus the rate of destruction of the cancer cells. In the arrow notation, I'd have nothing goes to cancer cells and the rate. Cancer cells go to nothing, the death and the death rate. So the creation rate and the death rate separately. If I write dN by dt equals some mess, I don't know which parts of that mess were the birth rate and which were the death rate. I can't separate those two things out. And so the arrow diagrams give you more information than the ODEs. On the other hand, and I can always write, if I write arrow, nothing goes to A, semicolon, rate, that's equivalent to writing dA by dt equals that rate. So I don't need to have an explicit declaration of an ODE, but if I'm going from a paper that has ODEs and I want to write something that looks the same, if I write K2 prime equals 3 times K1, that prime is d by dt. And so let's try doing that. Let's try K2 prime equals 3 times K1. And run that. And notice there's a comment that you have to specify the initial value of K2, because if you only specify the time derivative of K2, that doesn't give you the value. You have to have the initial value and the time. If you go back to ancient history, prime was used as a notation for time derivatives. Physicists are used to that as a notation for time derivatives. So K2 prime is equivalent to writing d by dt of K2. So did that work? When you do the plot, is there something different about the plot? It plots K2 now. So r dot plot plots everything that is a variable. And when you declare K2 prime equals something, then K2 stops being a parameter from the point of view of antimony and becomes a variable. And I noticed in several of the models that people downloaded, it did have the K2 prime syntax, the K2 prime equals something, I saw that in several of the models that you downloaded. And I also saw the colon equals syntax in several of the models that you downloaded. So having done those downloads, now we've done a little bit of discussion of the syntax of how it works in Antimony to do that. Okay. And so if I don't want to plot the K2 prime, then I'll have to do the column selection, or I'll have to use a different plot function. So I could use, for example, in the simulate, I could say only output A and B. So try that. To select the columns to display. Did that work? All right. Well, I think, let's see, I don't want to launch into something elaborate for the rest of today. We'll do this next time. We'll do the toggle. Were people able to get the basic toggle circuit to run for the homework?
I don't know, right?
Do you want to show what you got? Maybe we could do that.
Can't remember which one that was.
I think it was E. Because I have a fairly elaborate analysis of that circuit. That to be okay, let's put that up on the screen here. All right. Yeah, so it looks like you've got pretty much everything. So let's go to the very top there, where you define the model. Okay, so you have auto activation. No, wait a second. So this is not quite okay. So that's what did we do there? So this is
one based off the equation.
so you have 1 over Kn plus, so you have the rescaled forms of the Hill functions. And I have to mentally try to reorganize that. That is equal to a to the n over k to the n plus
A to the N plus B to the N.
a to the n plus b to the n. Okay, so that's an activating Hill function at A and inhibiting Hill function in B. So that's your cross inhibition and your self-activation for A, okay? And for B, you have the flipped one where you have, it's symmetrical, but the flipped one. Okay, so that looks fair enough. And then you have auto production of A, it's okay. And you have some first order degradation of A. And then you've got the pulse circuit, that at syntax that we were just talking about there. Okay, so that looks, I'd actually have to look at the homework assignment to remember which form of the Hill function I use, because in my analysis in my class notes, I used a slight. You made the comment that when you have two signals that come together, you have to decide how to combine them. And Herbert has made one decision about how to combine A and B. And in my class notes, I've made a different decision. They both combine activation and inhibition so that the more activator you have, the more you get. The more inhibitor you have, the less you get. But Herbert made a slightly different choice about how to do that combination than I did.
Yeah, I think I kind of settled on like you want your activator on the top of that hill
Right,
function,
that's right.
and you like compressor just on the
Yeah, that works. That absolutely works as a way of doing it. One thing to think about when you are doing analysis of what are called incoherent networks, where you have two signals coming in and the signals are trying to do opposite things, is ask the question, if one of the signals is 0, the other one is 1, what is the output of the circuit? So in this case, if you have A equal A and 1 in this case is scaled relative to K, so you want what is 0 would be less than K and big capital K, and 1 would be much bigger than capital K. So in this case, if you have yeah, so here you have a situation which in the first one, you have both A and B go to 0. In the second one, A goes high and B goes low. In the third one, A goes and B both go low. So are these different values of K? What is the only?
for this top left one is small a b equal to k.
This is surprising me because normally you would expect that the long-term state, the A star state, is independent, should be the same. You should be able to get two basic states, that either A star is big and B star is small, A star is small and B star is small, A star is big and B star is big, or A star is big and B star is small. There should be 4 cases, but there should be only 4. And so the final values that you get should be, and I guess if I look at the third row, A star is always 9.96, which looks, B is 0. So that makes sense to me.
Could it just be an interruption?
The top one, A star equals 0.18, 0.10, and 0.09. It's possible you didn't wait long enough for the thing to equilibrate and that it would have gone to 0. But what's a little bit surprising here, given that the system is symmetrical, is that you could have a situation where both A and B go to 0. You would that because the circuit is cross inhibition, you would expect that if A is stably high, B is stably low. If A is stably low, B should be high.
And that's why I didn't do, like I switched it to try and find where it was, but originally I didn't.
Right.
It's gonna. It seemed to just depend on concentration of A. This is why I was like kind of confused.
Okay, so what if you make the initial value of B non, okay, no, but you're starting with B, initial value of B is not 0. Yeah, the final state should depend only on whether A, initial value of A is bigger than B or not. If A0 is bigger than B0, the system should go to A being high and B low. if A0 is less than B0, the system should go to A low and B high. If A0 equals B0 at the beginning, then the system gets into a race condition, and then depends on the details.
Yeah, I got B always going to 0.
Yeah. So in that case, there is some error in the model definition. So let's take a quick look at the model definition, and then we're out of time, although I'm happy to meet with you one-on-one on that one.
To redo it as
Yeah, that looks fine. Those look okay. So something else is wrong somewhere. Where? Why don't we add a basal metabolism? So for R1 and R3, add a small value, yeah, B1, and make those non-zero. See if that helps. Yeah, so I'm usually pretty good at catching these things, but tonight I'm tired and I may not be able to do it.
it's, I don't know if there's something weird.
But definitely the final result is not. If B is always going to 0, then there's something wrong either with your initial setting of B, which I don't think is the case. Let's see, hold on a sec. Let's look at this for a sec. So, oh, I know what it is. So no, no, it's not that. So KDB is 0.6 and KDA is 0.2. So make them equal. Make them both 0.2, or is it, well, 0.6, they'll probably both go to 0 now. Yeah, you've got too much decay, so you're killing your oscillator. So make them both 0.2. There we go. Now you got it. Yeah, that's it. That was it. So you had built an asymmetry into the system where you had B decaying very rapidly.
Yeah.
And that decay was so fast that it was killing the B, no matter what you did.
Okay, yeah, I thought that was in the like formulation you had, and I think I picked up on that as the reason why I was always going to 0, but I didn't consider like what would happen in 30.
And actually, that's one of the specific exercises we were going to do in the later part of this class originally, was to analyze as you change that decay rate, what happens. And there is a bifurcation. If the decay rate is too low, both A and B go high no matter what. If the decay rate is too fast, then both A and B go to 0 no matter what. Right. Okay. So I think now you're not. So that show that diagram there for A. See, there you see if A is in the middle, it sits at half, it sits at 5 until there's some noise that throws you one way or the other. If A is above a half, it goes to 10. If A is below a half, it goes to 0. And so now if you reran that one with the 0.2 value, B will do the same thing. So rerun that cell, rerun that cell. Okay. Well, now we've got the both. We've got somehow we've got that that the decay rate to be too big for both. So we'd have to play with that a little bit. Where is the B? Something else got it. Oh well. I don't want people. I don't want to keep people too late, but
Well, my homework is gonna be wrong.
that's okay. We could since we didn't get to the point where I was saying, if people want, do you want to take another week to finish that homework? I mean, you could say no. No is an allowable answer. But what I would suggest is, given what we've discussed today, if you're happy with your homework, then for next week, read about. We're not going to get to parameter fitting next week. Do read the sections on. Stability analysis and phase plots, which were 12 and 13. Are the ones that cover what we need for the analysis. They cover bifurcations at phase plots. So take a look at chapters 12 and 13. And I appreciate people's patience. Sometimes things work the way that you hope, but sometimes they don't. So, but next week we'll do the toggle exercise in a little bit more detail. We talked a little bit about bifurcations last week. So these are the stable steady states as a function of one of the parameters, in this case, the degree of cross inhibition. And we'll talk about how to plot those. This picture here on the right is what's called a orbit plot or a phase plot. And what we've done there is we've started with a whole variety of initial conditions. This is the toggle, and what we have on the x-axis is the value of A, and on the y-axis is the value of B. And each one of those lines is, if I start on that line and I run the simulation forward, that's the trajectory of that line. I see that I could either have a large value of A and 0 value of B in this case, or a large value of B and a 0 value of A. And if I start with A bigger than B, which is the lower right-hand side of this box, I always wind up at that point there. If I start with B bigger than A, I always wind up on that upper left point there. If I wind up with A equals B, I have that line that separates this into two halves. And I don't actually know what happens based on that. I'd have to do more analysis. And so this shows me what are called fixed points. There's one fixed point, large A, 0B. There's one fixed point, large B, 0A. There's what's called a separatrix, that line down the middle. Everything that starts on the upper left goes to that upper point. Everything that starts on the bottom right goes to that lower point. These colored lines that are very faint, probably a little bit hard to see, are called null lines. And if we were going to do dynamical systems analysis in more detail, we'd spend quite a bit of time on how to use those. They're a convenient way of doing essentially analytic analysis of this kind of thing. I didn't think that we'd probably go into null lines much in this year. Calculating them numerically is a little bit tricky, and I have a code notebook that'll do that for you. so that you don't have to figure out how to do it. And I should say that I didn't actually come up with that code notebook. One of the students in this class a few years ago was the one who came up with it, because I struggled with it and couldn't come up with a way of doing it. All right. So for today, I think that's it. Thank you for being patient. Thank you for trying things out and showing things on the screen. I really appreciate everybody being willing to participate. And it's fine if things don't work. So, I mean, it's okay to debug things together.